Howdy folks, thanks for checking in to Mr. Ulrich's LandOfBiology.com. I am Mr. Ulrich. In this little notes cast, we're going to be talking about fire. Actually, we're going to be talking about the release of energy and how that relates to cells and cellular processes. Since we're going to be talking about interactions of matter and energy, we should go back to some basic chemistry just to review. These are symbols, and symbols represent the different elements. When we start putting different elements together and covalently bonding them into these types of structures called molecules, we use chemical formulas to write those down. When we start taking different molecules and putting them in the right environments so that they react and create different molecules with different chemical properties than the ones that they had when they started out, of course, now we're starting to talk about chemical reactions, and these are described using chemical equations. So we're going to be talking about quite a few different chemical equations. When it comes to measuring energy, I can't really hand you a pile of energy and say, here, how much is this? But what I can do is I can measure its output. Since energy is the ability to do work, if I measure how much work has gotten done when I expend some energy, I get an idea of how much energy that I have. Now, for us in biology, we typically talk about calories, capital C, which are actually kilocalories. You'll see that in the lab that we do. But there are plenty of other units, many that you'll talk about when you get to chemistry and when you get to physics. But it would be kind of weird for us to describe the amount of energy there is in a candy bar by measuring it in tons of TNT. That would be a pretty small number. So what we use are calories. Calorie, small c, is the amount of energy needed to raise 1 gram of water 1 degree centigrade. If you're writing that all out in notes, here, just write it like this, much easier and shorter. So 1 cal, 1 gram of water up 1 degree centigrade. So if I'm going to measure this, what do I need? I need water, and I need a thermometer, basically, are the two most basic things, and I've set up what's called a crude calorimeter. Now we're going to set one of these up in class in a lab, and we'll measure some calories of some food. But basically how it works is you take a fuel, a cheeseburger or a block of wood or a bubble of hydrogen gas, and you burn it. You oxidize that material so you release the energy, and that energy is released sending molecules banging into one another and getting the molecules of water to move faster and bang into one another. With my thermometer I can see how much more banging around those water molecules are doing as a result of the release of the energy from whatever. The ultimate energy source of all things is the sun. We are all, for all intents and purposes, solar powered. Even if we're burning coal to generate our electricity, that coal really represents old swamp plants that took the sun's energy and used that to fix inorganic carbon into organic carbon, which we then turn around and burn. I mean, it's just millions and millions of years later, that's all. Be nice if we could use light's energy directly, but we can't. Thankfully, there are those autotrophs, those swamp plants, and those potato plants that can take the sun's energy and use it to make fuel molecules. So they're the ones who do the magic of converting light energy into chemical energy. It's not magic. It's really it's biochemistry, and in this case, it's photosynthesis. To look a little closer into photosynthesis, why do plants do it? Of course, the big purpose is to generate energy, and in this case, they also need to have some sort of a portable, stable storage molecule for that energy. Some of it's going to go into ATP, which we'll talk about later on in the unit, but that molecule is very temporary and only works in the cell. So they can take some of the energy and use it, like I said, to fix inorganic carbon into glucose. So where does this take place? The chloroplast is the organelle. What fills the chloroplast, and there's the mnemonic device, it doesn't fill it completely, but what's in the chloroplast is that compound chlorophyll, or chlorophylls. There is more than one. But the job of that pigment is to take the energy from light and use that to break apart the water and energize the electrons using photons, so that the cell machinery can put that carbon together, that carbon dioxide, that inorganic carbon dioxide together into glucose, store that for later on, also make some ATP to do some stuff for the plant. But it does make oxygen as a waste product. It's not the goal, and for a long time that oxygen was actually toxic. So the light energy is what is used by the chloroplast. Well, I guess now would be a good time to compare and contrast autotrophs and heterotrophs. Autotrophs, of course, means self-feed. Those are the things that use photosynthesis to make their own fuel molecules. They do so by converting that light energy into chemical energy. Other organisms, like you and me, have to consume other organisms for their food molecules. We can't make our own fuel molecules. So we are heterotrophs. We have to feed on other things for our chemical energy. Hamburger. So how do we actually utilize this chemical energy once it's consumed either as fuel molecules or made as fuel molecules by heterotrophs? Through that life process of cellular respiration, the release of usable chemical energy from the breakdown of larger molecules, usually glucose. This takes place, most of it anyway, takes place within the mitochondrion. We can compare this to combustion. That's why I have this burning picture behind us. Combustion is a really quick chemical process, makes heat and light, but it's still the release of energy from the breakdown of larger molecules. And when we're burning wood, it is essentially burning glucose. The starch molecules are made out of glucose. Those lignin molecules are made out of glucose molecules. So really, cellular respiration is kind of an enzymatically controlled combustion, enzymatically controlled oxidation. The difference, of course, is that it releases the energy in a different form. It doesn't release it in the form of light and heat so much. Though you certainly wouldn't run your battery down on your cell phone in class using it there. Periodically, they do need to be, of course, recharged. And just like cell phones, your cells need an energy source, a temporary energy source, to move that energy from the storage molecule, glucose, to a form that can be used by the cell. Glucose has quite a bit of energy in it compared to what needs to be done by the cell. So what our cells do is they take that energy that's in glucose and they transfer it into another more usable, more readily usable compound called adenosine triphosphate, or ATP. The way we make ATP is you take a molecule called adenosine diphosphate. The big block there is an adenosine, or represents one, and it has two phosphate groups attached to it. Phosphates, having two phosphate groups right there makes it pretty unstable, but it can be bonded together. And then we could take another phosphate group and stick that on there and make our compound called ATP, but that phosphate is not bonded real well onto that last phosphate. It really wants to break off. It actually represents quite a bit of energy to just hold it on there. And because it wants to pop off so easily, it makes it a really nice candidate as a molecule to transport bond energy from one place to another. So as you go through cellular respiration, the process is to ultimately bond P's onto ADPs to make ATPs. And then when you do things like, say, active transport, you break one of those P's off of ATP and use that bond energy basically to change the shape of something, usually a protein, to cause it to move or do something. So that cellular activity requires the breakdown of ATP into ADP and P. And so this ATP cycle constantly goes back and forth between ADP and P, and then charge it up into ATP and then discharge it, doing cellular activity back to ADP and P. Photosynthesis is taking place within the chloroplasts of mostly plant cells. We need to be able to associate cellular respiration with the slipper structure, or the hot dog with the mustard on it, that structure that we described as the mitochondria, the powerhouse of the cell. Now we're going to spend more time on this in following classes, but right now we need to be able, like I said, to associate that mitochondria with cellular respiration. We'll talk more about aerobic versus anaerobic cellular respiration and how the process really does start in the cytoplasm anyway, but like I said, we're going to talk more about that next class. Thanks for checking in to Mr. Ulrich's landofbiology.com. I am Mr. Ulrich. If you have any questions, comments, concerns, cliches, cataclysms, or any other chrysanthemums, make sure you drop me an email there at the address on the screen. You can also go to MrUlrich'sLandofBiology.com if you got here through my YouTube channel or by some other link, and get the note sheet and any other associated nonsense that might help this whole process. Other than that, we'll see you in class.